In this lesson, I am introducing enzymes and how they work. We're going to look at enzymes and their function throughout this course, so it's important to have an understanding on the basics. So first of all, an enzyme is a biological catalyst. The catalysts speed up a chemical reaction. So this is one that just naturally exists in cells. So that's where the biological part happens, and it is speeding up a chemical reaction. So these chemical reactions that are happening in the cell would theoretically still happen, but just at a much slower rate. And we're going to look at some really important reactions again throughout the course where without an enzyme, we just wouldn't be able to have functional cells. Over here we do have a bunch of examples of enzymes and just some that are very common in our cells, very important. So we have glucose oxidase, for example, and I'd just like to show you their 3D structures on what they look like. This one here, we can see some alpha helices. You can see them a lot in these ones. So this is alcohol dehydrogenase, so this is breaking down alcohol in the cell. Urease, which is important in urine production. And just go through and look at them. You are not expected to know these in any way. I'd just like to show you their 3D structures. Okay, a catalyst, again, is any substance that speeds up a reaction without getting consumed. So there's probably some labs that you may have done in grade 10 with manganese dioxide, which acted as a catalyst to break down hydrogen peroxide, and it wasn't consumed. So it wasn't involved in the actual reaction, but it was involved in making the reaction happen. Most enzymes are proteins. Not all proteins are enzymes, but most enzymes are a protein, biological enzymes. It does not get consumed. It does not change the products of the reaction. So again, it's not being consumed. Part of the enzyme doesn't become a product. The enzyme is intact in the same way before as it is after the reaction. Typically, a cell has about 4,000 enzymes in it. So very, very important structures. And each enzyme has a unique 3D shape, so as I've shown some examples here, and this determines which reaction it catalyzes. So its shape is very important. And we talked about the shape of proteins earlier in the unit and how that folding in the shape is what gives its function. So why do we need enzymes? So some chemical reactions need to overcome energy barriers. So there's some sort of energy problem, if you will, that we're going to talk about a little bit later in the next unit, where energy is needed to overcome the reaction, and the enzymes will lower that need of energy. Enzymes will bind specific reactants to lower that energy barrier. So they allow, and we'll talk about this again later, they'll actually bring reactants together to make it easier for that reaction to happen. Less energy will be needed. And it allows the reaction to proceed quicker than it would without the enzyme. So you can get more reactions happening, for example. Definition that you need to know, and I will use it from here on out. Substrate is the substance that is recognized by and binds the enzyme. So we have our enzyme. Whatever that enzyme is binding to is called the substrate. So an example, we have catalase. So catalase is a tetrameric protein. So tetra means 4. So it's a tetrameric protein, meaning it has 4 subunits. And this is its 3D structure shown here. Catalase aids in the breakdown of hydrogen peroxide. So in grade 10, we used manganese dioxide, which is just a chemical that we order that we could use in that reaction. In our bodies, we have an enzyme called catalase, and this aids in, like I said, the decomposition or the breakdown of hydrogen peroxide. Hydrogen peroxide is toxic, and it's a byproduct of metabolism. So it's actually produced in the body, so the body needs a way to break that down. And here is the reaction shown here. So we actually have hydrogen peroxide being broken down into water and oxygen, which is actually exactly what happens in that grade 10 lab where we had manganese dioxide up here. Enzymes of any sort or any catalysts are drawn above the arrow because that indicates that it's not actually a reactant or a product. It is a catalyst of the reaction. So this is where we would put the enzyme name. So enzymes are very specific to the substrates that they bind. So each enzyme catalyzes the reaction of only one type of molecule or group of similar molecules. So they are extremely specific. So shown here, again, we have catalase. Look at the massive size of this structure compared to hydrogen peroxide. So here's the enzyme here, and here is our hydrogen peroxide. This is pointing to the active site here. So this is another definition that we'll talk about in a little bit. The active site is where the substrate binds. We don't draw enzymes like this. We draw them in a nice little primary, I'd call this primary shapes. So this is the enzyme, and we have the substrate shown here, and this is where we would have the active site drawn in. So it looks very much like a lock and key shape where you can visually see where that substrate would be binding in the enzyme. So enzymes, as I said, are much larger than the substrate. And the active site is the region on the enzyme that interacts with the substrate. So again, visually, this is more what it would look like, but this is how we represent it. Usually, a pocket or a groove that forms when the protein folds into its 3D shape is what creates the active site. Shown here, this is so I had said earlier about it looks like a lock and key, and that's actually what the old theory was. But the new theory is the induced fit model. So they're similar, but a little bit different. So we're just going to talk about the induced fit model. In university, you will learn more of the background or the history of how knowledge came about enzymes. We're just going to talk about what we know. So our induced fit model is telling that the enzymes are not rigid molecules but flexible. And we can think about that when we have our protein folded, it will have some ability to change a little bit of its form. It'll be more pliable. And as it binds the substrate, the enzyme actually changes shape, and this changes its conformation. So it allows a much better fit for the substrate. So we see here, we have our enzyme here, and we have our substrate. And our substrate is made up of two sections, if you will, and it doesn't really matter so much, but it's in this case, we're going to have a breakdown of a molecule. So you can almost think of hydrogen peroxide, okay? And it would be binding in this active site here. So it looks like it fits pretty good, but there's just a little bit of a gap, if you will, in this active site. As it binds, there's going to be a bit of a shape change in the enzyme complex that's holding the substrate. This allows for a much stronger, tighter fit, and the enzyme will then do its job, and in this case, it's breaking this substrate apart. There's times when it's actually taking in two parts and bringing them together, so that can happen as well. So the active site becomes more precise in its ability to bind its substrate. After it is bound, it changes its shape a little bit, and it has a better hold on its substrate. So that's the induced fit. So binding to the substrate induces a change in the shape of the enzyme. So there's this thing called a catalytic cycle, which sounds a lot like that car part. Catalytic converter. So first, the enzyme binds to one or more substrates, and again, it depends on the type of reaction. So with the decomposition of hydrogen peroxide, there's only one substrate. There might be two substrates where it's bringing it together, which would be a synthesis reaction. This forms an enzyme complex substrate. The enzyme then converts the substrate or the substrates into one or more products. The enzyme can rapidly bind to other substrates, catalyzing the same reaction over and over. And the rate at which an enzyme catalyzes the reaction varies. So it depends on the enzyme and the environment in which the enzyme is in. And we're going to talk in class tomorrow. You're going to do a little jigsaw activity where you get to be specialists and teach the rest of the class about different things that affect enzyme functionality. So let's look at an example, and this is right out of the textbook. We have beta-galactosidase. Beta-galactosidase. Its short form is beta-gal, and that's what we're going to call it. It's also called lactase. So this hydrolyzes, so this breaks down lactose. And we looked at a little bit at the structure of lactose. Lactose is produced by glucose and galactose. So it's a disaccharide. So this is a disaccharide. So we have glucose and galactose, and this is lactose. So lactose is going to be broken down by beta-gal, and let's look here. So we have beta-galactosidase, beta-gal, and it binds lactose. And it's shown here as two subunits because lactose is made out of glucose and galactose. This is the enzyme substrate complex here. Water is added, so this is a hydrolysis reaction. We did this in our model building lab, where water is added to break apart the lactose to produce glucose and galactose. So we see our products are being removed, and we're left with our empty enzyme with its open active site. And the same will continue again. So we'll have lactose binding to the active site of beta-gal, shown here. Water is added, the bond is broken, glucose and galactose are released, empty enzyme, and again. And it just goes as long as the enzyme is still there and there's lactose to be broken down, water will always be available, this reaction will keep going. So just to make your lives a little more complicated, there's two terms that we add to this. So we have coenzymes and cofactors. So first we're going to talk about coenzymes. So in this diagram, we have what's called an apoenzyme. So this is the enzyme structure with just the enzyme. There's these things called coenzymes that need to bind in the active site in order for the substrate to bind. So it's a non-protein portion. So remember, it's almost like a prosthetic group. Okay, so we have and this is called an apoenzyme because the enzyme isn't functional without the coenzyme. So it's just, again, another term. Sorry, guys. Okay, so we're going to have the binding of the apoenzyme with the coenzyme, and specifically this coenzyme will bind in the active site. This is important for you to keep this separate from the next definition. Okay, it's the one way that they're different. And then once this coenzyme is bound to the active site, then the substrate can bind. And it's called a holo enzyme, which is a whole enzyme. And this is active. All right. So the coenzyme often derived from water-soluble vitamins, so not K-A-D-E. So it's important to remember KD are the fat-soluble ones. So those are actually the ones that you can consume too much of. Typically, if you consume too much of the other vitamins, you just pee them out and it's not a problem. But K, D, E are the ones that can build up in your body. Many shuttle molecules from one enzyme to another, and they often bind loosely to the active site of an enzyme and aid in the substrate recruitment. We have nicotinamide adenine dinucleotide, NAD+, we're going to learn all about in the next units. It's a derivative of vitamin B3. It acts as an electron carrier during a number of biochemical pathways that we're going to talk about. Okay, similarly, we have cofactors. The difference is a cofactor doesn't bind the actual active site, it's just binding near the active site. So it's a non-protein group, again, and it binds very precisely to the enzyme. It's needed by some enzymes for catalytic activity, and it often will be metals like iron, copper, zinc, or manganese. Magnesium plays a huge role in cellular respiration in mitochondria. They do not bind the active site of the enzyme, so this is how a coenzyme and a cofactor differ. So a coenzyme would be in the active site, whereas a cofactor is just outside of the active site. So something to keep in mind.